Dear students, let us study the different forms of DNA. So, before knowing the different forms of DNA, let us know different types of DNA. So, DNA, you know this, you have studied in the last sem the structure of the chromosome. So, eukaryotic organisms, DNA is present inside the nucleus. It is a part of the chromosome. And also you will find some DNA molecules also present inside the mitochondria and chloroplast cell organelles. So, nuclear DNA, it is you can see it is the part of the or constituent of the chromosome and which carries the genes. So, the nuclear DNA is diploid. It is you know that chromosomes, haploid number of chromosomes contributed from the sperms and haploid number from the daughter going to result diploid zygote. So, you will find it is DNA what is there in the nuclear DNA is inherited from the both the parents. In human, the chromosomes contains 3 billion nucleotides. The mutation rate of this nuclear DNA is very less, it is less than 0.3%. Coming to the mitochondrial DNA, it is there are some circular DNA molecules are present inside the mitochondrial matrix. So, in case of human, it is this circular DNA contains 16,569 nucleotides. This mitochondrial DNA is haploid because it is inherited from the mother. It the egg cell, whatever the mitochondrial DNA, it is the present in the egg cell, it is a part of the zygote later. So, it encodes the mitochondrial DNA encodes the proteins that are specific for the mitochondrial that is the you know the mitochondria where you can see the metabolic pathways. It is concerned with the energy producing pathways of the mitochondria. The mutation rate is generally higher in the when compared to the nuclear DNA. Then chloroplast DNA, the found in the photosynthetic land plants, is a circular genome similar to mitochondrial DNA, and it contains it is 1,20,000 to 2,47,000 nucleotides. They contain genes that are involved in the photosynthesis or else chloroplast activities. Coming to the different forms of DNA. So, there are DNA occurs in different forms A, B, C, D, E and Z forms. Before going to see all this, so let us know about we have studied already the Watson and Crick model of DNA, that is the DNA double helical model, double helix it is called. It is nothing but B form of DNA. Under normal physiological condition, inside the cell, the DNA occurs that is in the form of B form. So, where you will see you have studied already the B form. The two strands run in opposite direction, anti-parallel. DNA structure is asymmetrical and you will find there is a major groove and there is a minor groove, you have studied already. So, this is because the glycosidic bonds of base pairs are not diametrically opposite to each other. So, you will find in the base pair, you will see here. So, this is this glycosidic bond here and in here, that is the bond between the base and the sugar. So, it is not in the same level you will find. So, that is why it is causing a major groove and a minor groove. In each in one turn there are 10 base pairs and the length it spans about 3.4 nanometer or else 34 angstroms. This is one complete turn, 360-degree turn. So, this contains 10 base pairs in the B form. The distance that is why the distance between a a adjacent nucleotide, what it is? It is 3.4, you can see angstroms, or else 0.34 nanometer. So this backbone of this bDNA is that is the sugar and phosphate. They are linked by the you can see the phosphodiester bonds, and the core region, this is what you can see, the base pairs. So this is a nitrogenous base, you can see. So this is the B form, that is what the Watson and Crick proposed, the double helical model. So most of the DNA is the classic Watson and Crick model, and it is called bDNA. This is the bDNA. So bDNA commonly found consists 2 helical polynucleotide chain which runs in a antiparallel fashion. So which contains a major groove. It is the major groove, it is about 22 angstroms depth, 22 angstroms wide, and the depth is about 8.5 angstroms. So then a minor groove, it is about 12 angstroms wide, and depth is about 7.5 angstroms. So the major groove occupied by the water molecules. So this the major groove is also, there is a binding of some transcription factors, and then proteins, and all these. That's the site where you can see the major groove. There is a transcription factor is bound here in this cartoon. So DNA is not uniform and can vary under different physiological condition. So different forms of DNA like B, A, C, D, E and Z have revealed after X-ray diffraction analysis of DNA. So the B form changes into A form or else Z form. So you can see the most of the time in normal physiological condition, so the DNA occurs in the form of B form, what you have studied, the Watson and Crick model. So what are the factors affecting or bringing this conformational changes from B to A or B to Z form? So the factors bringing structural diversity of the DNA or changing its structural configuration. So these are the factors. The number one, the hydration level. So the humidity and the ionic concentration. Hydration environment favors the conformational changes in the DNA. So if it is the 92% of humidity or else hydration, the DNA occurs in the form of B form. So then if it is 75% of hydration level, then it occurs in the form of A form of DNA. And the high salt concentration, high salt concentration, it changes into Z form. So these are the factors which is bringing the structural diversity of the DNA or different forms of DNA. So the base sequence, that also decides the shape, size, coiling pattern and structural properties of the DNA. Then the quantity and direction of the supercoiling. So you can see this is the two strands opened up, and this is a double helical structure, and it is the supercoiled you will find. So this also affect the form of DNA. Presence of modified bases. The bases, usually normal bases, adenine, guanine, cytosine, thymine, you know that DNA bases. So sometimes these DNA bases are methylated, so they are modified bases. Then the presence of metal ions. So the metal ions, or else you can see the polyamines, so the low molecular weight bases. So that is if it is present in the solution, which changes the DNA B to different forms. The presence and binding of proteins. A protein binds with the one helical conformation of the DNA and changes its structure into different forms. So some protein binds to the B form and changes it into the A form. So a protein can bind to the B DNA and change the conformation to A or Z form. So here in this cartoon you can see this is the A form. So if you take the section, or else if you look from the top, you will find like this. This is the B form. This is the normal physiological condition. This is the Z form. So what are the differences you will find? So this is B DNA, normal one, A, C, D and Z. So this is the B, A, you will find C, D and the Z. So double helix. So the B form right handed and A, C and D all right handed. The Z form is left handed. Base pairs per turn, one complete turn, 360 degree turn, you know that. So it is you can see this is 10 base pairs, whereas in A it is 11 base pairs. In the C it is further you can see the size is reduced. It is 9.33 base pairs. D 8 base pairs. Z is little extended. It is 12 base pairs. What is the size, the diameter of the? So it is this one is 2 nanometer or 20 angstroms. A is you can see it is wider, 2.3 nanometers. C is little it is 1.9 nanometer and Z is 1.8 nanometer. What is the rotation angle per turn? So it is the angle per turn is 360. So that's why 1 base pair 36 degree, 10 base pair 360 degree. Here 32.7 degree, 38.6 degree. So it is in a reverse order, minus 16.70. So it is minus 30 degree. So it is the left hand double helix, that's why. So this is what you can see, right hand double helix, the left hand double helix you can see in the case of Z DNA. If you treat with the EDTA, then the left hand will change into right hand. So then the helix axis located. So where exactly the helix axis is located? So this is in the case of B DNA, it is in between the base pairs. Helix axis is located in the major groove. This is in the minor groove. So this is in the base pairs. This is in the minor groove. So what is the nature of the major groove? It is in the case of B DNA it is wide, and in the A DNA it is narrow and deep, and in the C DNA it is wide and less deep. And we don't know about this because it is not there is no much difference between major and minor groove. It is not forming major and minor. So in the case here it is major groove is flattened, you will find it is flattened. Minor groove is narrow in this is minor groove, shallow in the case of A form, and narrow and deep in the case of C form. We don't know about this because there is no much difference really. So it is very deep in the case of, this is what you see, it is a minor groove. Then physiological conditions, here 92% of humidity, low salt condition, B form. You can see in the solution rich in potassium and sodium, and 75% of humidity, A form. Then 66% of humidity and in the presence of lithium and magnesium ion, C form. So we don't know the conditions here in the D form, when D form occurs, and high salty solution, then Z form. So you can see here this is a hydrated B form, 92% of humidity. When less hydrated it turns to A form, you will find. So the left handed Z DNA here you will find can exist within the regions of the B DNA. This is the B form. In between you will find a Z form, and here the B form. So that is what you can see in living cell. Most of the DNA is in the mixture of A and B DNA conformation with the small regions capable of forming Z DNA. So these the the form, the structural conformation changes. You can see most of the time it will be B. It may in between you will find the Z, or in between B form there will be A. So this is what you can see. This may be the conformation may change from B to Z in certain region. So what is the significances of this? So the local structural transition from the common B DNA conformation to other DNA forms, it is very important, because such a transition in certain sequence or in the certain elements of DNA can be induced by changes in the environment. It may induces these conformation changes. Some certain protein binding bring the changes, and some helical, super helical for transformation bring these changes. So alternative DNA structures may exist in prokaryotic and eukaryotic cells. All these forms you will find even in prokaryotic and eukaryotic cells. They are involved in the replication process. You can see this A form brings the replication, induce a replication. So likewise they may induce the gene expression, the recombination, and also mutations. So that's how the Watson and Crick model, what you have studied, it is for the B form. So the DNA exists in other forms also, A, Z, Z, D and other forms. Thank you.